Hi everyone, welcome back to my educational channel on biology. I'm teacher Janet. In this video, we're going to discuss effect of light intensity on the rate of photosynthesis. So we'll be discussing this experiment in detail, all right, and we must know how to set it up and then know what are the MVs and RVs and so forth. So here I've prepared a very good video for you, all right, and based on my vast experience in preparing practical tests, and also we are going to discuss paper 3 answering techniques, right? So you can revise the techniques and be able to apply them to the question that's given to you, right? So that will help you to score high marks in the exam. So without much delay, we will get started, right? So here's some background knowledge about photosynthesis before we begin. The word equation for photosynthesis is water plus carbon dioxide with the help of light energy absorbed by chlorophyll in the leaf produces glucose and oxygen. Our acronym is WECC, WEC, arrow, GO, go. Okay, W for water, E for energy, which is light energy, C for carbon dioxide, C for chlorophyll, and then GO stands for the products, glucose and oxygen. So this oxygen released is a byproduct of photosynthesis, and we can observe it in the water plant when the water plant produces the gas bubbles. The gas bubbles are the byproduct, that is oxygen gas. Okay, right. It will be released as bubbles. Now, factors that affect the rate of photosynthesis. There are 4 factors that are important here: light intensity, carbon dioxide concentration, temperature, and even color of light. Now, for light intensity, we know that light energy is needed for the photolysis of water in the light-dependent reaction, LDR, light-dependent reaction of photosynthesis. So light provides the energy for the splitting of water molecules, all right, to form the hydrogen ions, hydrogen atoms in the end, and the hydroxyl ions. So now let's look at the graph. The graph is a graph that goes upwards and then it levels off. Okay, so as the light intensity increases, the rate of photosynthesis increases until a certain level. Okay, then it levels off because of the limiting factors. Now, carbon dioxide concentration. Carbon dioxide is important because it's the substrate, one of the important substrate for photosynthesis. Okay, so as again the graph is similar to that of light intensity. As the carbon dioxide concentration increases, the rate of photosynthesis will increase until a certain level. Okay, temperature affects the photosynthetic enzymes which are used to catalyze photosynthesis in the plant. So at high temperatures, for example, these enzymes will be denatured, and the optimum temperature for photosynthesis in plants is around 25 degrees Celsius to 30 degrees Celsius. It's lower than for humans. Okay. Now, for temperature, the graph is a bell-shaped curve like this. Okay, it goes up, and then there's a maximum point. That means that is the optimum temperature for photosynthesis to occur at the highest rate. And after that, it goes down. That means the rate of photosynthesis decreases as the temperature gets higher and higher, because at high temperatures, the enzymes are denatured, okay? And the structure of the protein molecules of the enzyme will be destroyed, will be changed. Now, color of light also affects the rate of photosynthesis. So red and blue light has been found to be easily absorbed by chlorophyll, okay, and other pigments. So it can be used in photosynthesis. But chlorophyll does not absorb green light. It reflects the green light. So in green light, the rate of photosynthesis will decrease, okay, because the plant will not get enough light energy for photosynthesis to occur. So here's an interesting thing to note before we start discussing the experiment in detail. For this experiment, there are 2 manipulated variables and 2 responding variables. Okay, now from the aim, you obtain the main variables. So this is the aim, to investigate effect of light intensity on the rate of photosynthesis. The effect of something, that something is the MV. The effect of MV on RV. Okay, so your MV is the light intensity, and that is called the main MV because it comes from the aim. And then the rate of photosynthesis becomes your main RV or main responding variable. Okay, but in the lab, when you carry out the experiment, what are you going to use to change the light intensity? Then that is your sub variable. You use the distance of light bulb from Hydrilla species. Okay, For example, in the picture here, this is the setup of the experiment. The Hydrilla species plant is inside the boring tube, okay, immersed in the sodium hydrogen carbonate solution, and here you have a light bulb. So if you change the distance between the light bulb and the Hydrilla species, you can move the light bulb nearer or further away. Okay, you can measure the distance using a ruler. It can be 10 cm, 20 cm, 30 cm, and so forth. Now, the nearer you move the light bulb to the Hydrilla species plant, then the higher the light intensity, correct? So distance of light bulb from Hydrilla species can be used to change the light intensity as the manipulated variable. So we call this the sub-MV, the distance of light bulb from Hydrilla species plant, because we use it in the experiment. Okay, now for the sub-RV, how do you measure the rate of photosynthesis when you carry out this experiment? Okay, so you are going to look at the oxygen bubbles released and count. So number of air or gas bubbles or oxygen bubbles released per minute is your sub-RV. That means the higher the number of the larger the number of the air bubbles, it means that more oxygen gas is released. That means the rate of photosynthesis has increased, okay? Because oxygen gas is a byproduct of photosynthesis. So your sub-RV is the number of air bubbles or gas bubbles released per minute. And from the sub-RV, you may be able to calculate the main RV, okay? So rate of photosynthesis equals to the number of air or gas bubbles released per unit time divided by the time, okay? That is your sub-RV, okay? So remember, main RV and MV is from the aim, and the sub variable here are the ones that you use in the experiment. Now these terms are terms that I coined up myself. You won't find it in the books, or it won't be mentioned by others. Okay, but it's an easy way to understand how the variables are related to each other. Important point to note. Concerning the sub-RV, the number of air or gas bubbles released in 1 or 2 or 5 minutes. All right, this part, you have to read the question paper, the statements there carefully. Is it asking you to count the number of air bubbles released in 1 minute, or in 2 minutes, or 3 minutes, or 4 minutes, or 5 minutes? Read it carefully and underline the number of minutes, okay? Don't assume that it's going to be according to what we have discussed in the exam, in the question here, or in the video here, all right? You have to read carefully how many minutes it is, which is the time taken for you to count the number of air bubbles, okay? So follow the question in the test paper, exam paper, okay, SPM question paper exactly, and do the question exactly as what they ask you to do. Adapt to the question, okay, and be flexible to adapt to the question, instead of just assuming everything is going to be like in the video here, or everything is going to be like what your teacher said or did, or according to the textbook. There may be slight differences, okay? There may be differences. So take note, read the question carefully. So now let's start by discussing experiment 2.3 in your biology TSSM textbook. Okay, this is the first factor, A, which is light intensity. So here our aim is to investigate effect of light intensity on the rate of photosynthesis. Okay, now that's your aim, to investigate the effects of light intensity on the rate of photosynthesis. So from the aim, we already know the main MV and the main RV. Okay, the effect of something, that something is the main MV. The effect of main MV on main RV, or the effect of MV on RV. Okay, so your RV is the rate of photosynthesis. So as you change the light intensity, then we expect the rate of photosynthesis to change also. Now, how about the hypothesis? How do you write a hypothesis? Your hypothesis must have three components: the main MV, main RV, and also the correct relationship. Okay, so in this experiment, we can use the format: the higher the MV, the higher or lower the RV. Okay, for example, for light intensity, we can say the higher the light intensity, the higher the rate of photosynthesis. Okay, that means as we increase the light intensity, we expect the rate of photosynthesis to increase, because the plant will receive more light energy for the splitting of the water molecules. during photolysis of water, okay, which is part of the process of photosynthesis. So the higher the light intensity, the higher the rate of photosynthesis. Now, variables. What is your manipulated variable? As we said, there are two manipulated variables. The manipulated variable, or sub-MV, is the distance between the light source and Hydrilla species, which is the plant, the water plant. And your main MV is the light intensity. Okay, you obtain that from the aim here. Okay. Now, for responding variable, the number of air or gas bubbles released in 5 minutes is your sub-RV. Okay, sub-responding variable. But the main RV is the rate of photosynthesis, as we have obtained from the aim here. Okay, as is stated in the aim here. All right. Then constant variables. The type and size of Hydrilla species plant, okay, or the type of plant, the concentration of sodium hydrogen carbonate solution, all right, which determines the concentration of carbon dioxide, because sodium hydrogen carbonate releases the carbon dioxide for the plant to use, for the water plant to use in photosynthesis. All right. So please note that you have to write concentration of sodium hydrogen carbonate solution. You have to write the word solution. Don't just write concentration of sodium hydrogen carbonate. It's incomplete. It's a solution, so you must write the word solution inside. So remember this word solution. The voltage of the bulb, even the temperature is a constant variable. Must keep the temperature constant. Okay, we only change the light intensity. Okay, now materials. Hydrilla species plant, 50 ml of distilled water, 0.2% sodium hydrogen carbonate solution. Okay, so the concentration of the sodium hydrogen carbonate solution is fixed at 0.2%. Apparatus, scissors, 60 watt bulb, meter ruler, stopwatch, paper clips, boiling tube, measuring cylinder, retort stand and clamp, thermometer and beaker. So here is the setup of the experiment. Okay, let's discuss how to set up this experiment. Firstly, we have to take and cut a length of Hydrilla species plant, maybe about 8 cm or so, okay, depending on the size of your boiling tube here. You're using a boiling tube. So first of all, get a length of Hydrilla species plant, and then attach a paper clip at the bottom of it. Or for some experiments, you can even use a plasticine. Okay, just wrap it around the bottom part of the Hydrilla species plant. Now, why is that so? It's a precaution. It's a precaution so that the paper clip or plasticine will weigh down the Hydrilla species plant right to the bottom here, and it will not float at the top. If it floats at the top, if the plant were to float upwards to the top, then the experiment cannot be carried out. You cannot count the number of gas bubbles released, right? So it must be weighed down to the bottom. So to ensure that the Hydrilla plant is fully immersed in the solution in the boiling tube. Okay, so after you have attached a paper clip to the Hydrilla plant, then fill up the boiling tube to near the top with the 0.2% sodium hydrogen carbonate solution to provide the carbon dioxide for the plant to carry out photosynthesis. Next, put the plant into the boiling tube and then clamp the boiling tube vertically to a retort stand here. So there's another retort stand on this side. Now next, your light bulb. So you're using a 60 watt bulb. So when you are ready, you just switch on the light bulb. Okay, and the light bulb must be placed at a distance of, for the first manipulated variable, it can be 10 cm or even 20 cm. Okay, let's say we start with 10 cm. The distance is 10 cm between the Hydrilla plant and the light bulb here. Right? So measure the distance carefully to be 10 cm. Now textbooks start with 20 cm. That's the same. So after you have measured the distance and placed the light bulb at the correct position, then you switch on the light bulb. Then wait for the plant to adjust itself to the new condition, okay, to the light intensity. So wait 1 or 2 minutes, because the air bubble or gas bubble will not be produced straight away at once. Okay, the plant has to adjust itself. But once you see the air bubbles forming or gas bubbles forming, all right, then wait for a few gas bubbles to float to the surface first. Okay, then you're ready to start counting the number of gas bubbles produced in 5 minutes. Okay, let's take the time as measure the number of air bubbles released in 5 minutes. Okay, for some cases it could be every minute or 2 minutes, every 2 minutes, and so forth, depending on the question. So read the question carefully. Now take 3 readings or 2 readings to obtain an average after that. Okay, so that means when you are ready to start counting, you have to start the stopwatch first. Start the stopwatch so that you can count 5 minutes. And then after that, as the bubbles are released, you count 1 bubble, 2 bubbles, 3 bubbles, and so forth. Okay, until your 5 minutes is up. Then you stop. Then you jot down the number of gas bubbles released in 5 minutes. Okay, after that, you repeat the experiment to get 2 readings and then obtain an average reading for the number of gas bubbles released per minute. Okay, now once you are done with this first part of the experiment, where the distance between Hydrilla species and the light bulb is 10 cm, then you are ready to go on to the next manipulated variable. Okay, where the distance between the bulb and Hydrilla species is 20 cm. Okay, and then you repeat the steps again. But before you repeat the steps, please remember to change the sodium hydrogen carbonate solution to a new one. Replace it, okay, with a new one, so that because the old sodium hydrogen carbonate solution would have released the carbon dioxide, and the concentration of the sodium hydrogen carbonate solution would have decreased, okay, after the first experiment. So you have to change the old solution with the new one, okay, before you start counting the number of gas bubbles for the next stage, for the next part where the distance is increased, right, to 20 cm, then 30 cm, 40 cm, and then 50 cm. So record your results in the table. Now let's look at the procedure here, and we're going to read through it. It's the same as what we have discussed just now. So first, attach a paper clip at the bottom of a stem cutting of Hydrilla species to weigh it down so that it can be fully immersed in a solution. This is called your precaution. Next, put the plant in a boiling tube containing 0.2% sodium hydrogen carbonate. Maybe you can pour the solution in the boiling tube until it reaches near to the top. Then you can start the experiment. So here the volume of the sodium hydrogen carbonate solution is not mentioned. Now, your 0.2% sodium hydrogen carbonate solution is the CV, the constant variable, or the fixed variable, FV. Okay, that means it's the concentration of the sodium hydrogen carbonate solution. That is the constant variable. Number 3, clamp the boiling tube vertically to the retort stand. Okay, and also get ready the 60-watt bulb. You have to clamp it also to another retort stand. Okay, then switch on the 60-watt bulb light with a distance of 10 cm from the Hydrilla species plant. So you have to take a ruler and measure the distance 10 cm between the bulb and the Hydrilla plant, first of all. Then only you switch on the light. Now before you switch on the light, make sure that you have given about 1 or 2 minutes, 2 minutes for the Hydrilla species plant to adjust to the light. And you have to see whether the air bubbles are released or the gas bubbles are released. If there are no gas bubbles, don't start the experiment. Okay, wait until gas bubbles start to form. Okay, they are released. Then you start your stopwatch. Now count and record the number of air or gas bubbles released in 5 minutes. Then after that, take 2 readings to obtain an average. Okay, so at this stage, your sub MV is the distance of 10 cm, and your sub RV is the number of air or gas bubbles released in 5 minutes. Okay, so as I put here, sub RV is the RV used in the experiment, that's measured in the experiment. Okay, sub MV is the MV that is manipulated in the experiment. Then next, replace the 0.2% sodium hydrogen carbonate solution in the boiling tube with a new one when you've finished the first experiment. So this is again a type of precaution, because if you don't change the solution, then the results will not be very accurate, because then you're using an old solution which may have less carbon dioxide, which may release less carbon dioxide compared to the first experiment. Okay, so you have to replace the 0.2% sodium hydrogen carbonate solution with a new one, so that the concentration of the sodium hydrogen carbonate solution is kept at 0.2%, right? Like before. Now repeat steps 3 to 5 at different distances between the bulb and Hydrilla species, that is 20 cm, 30 cm, 40 cm, and 50 cm. So this is your sub MV. Then record the results in a table. Discuss the paper 3 questions and also the answers, and I will be sharing with you the techniques of answering paper 3 questions well, okay, to help you score high marks in your exam, your paper 3 exam. So pay careful attention, and always remember that in the real exam you have to follow the statements or the MV and RV that is stated accordingly. So you have to adapt yourself to the question and read the question carefully, and don't assume that is exactly like what we have discussed here. Okay, so this video aims to teach you the techniques of answering paper 3, so that you can apply them to the paper 3 question and be flexible about how to write the exact answer that is needed for the exam. All right, okay, so let's get started. First of all, these are results that we obtained, all right, and we have written them down in the table. Now, distance from Hydrilla species to light source, you must write down the unit, cm. Okay, this is your sub MV. And here, what is the number of air bubbles released in 5 minutes? There are 2 readings. Okay, and finally, you take or find out the average number of air bubbles released in 5 minutes. That means you have to take these 2 readings, divide by 2. Okay, add up the readings and then divide by 2. Now, finally, you may be asked to calculate the rate of photosynthesis. How to calculate rate of photosynthesis? Well, in this case, the rate of photosynthesis is the number of air bubbles produced per minute time, per minute. Okay, the time here is in minutes. So, if in 5 minutes 21 air bubbles are released, then in 1 minute, how many air bubbles are released? So, 21 divided by 5. That's so simple. That means number of air bubbles divided by the time, you get 4.2 bubbles released per minute. Okay, of course, actually you cannot write 4.2 bubbles, but here in this case, it is just it is the result of this division. So we have to just take it as with the decimal place here. Okay. Now, by the way, what is the unit for rate of photosynthesis? It is bubbles per minute. Okay. And what's the unit for average number of air bubbles? It should be bubbles. Like 21 bubbles, 16 bubbles, 11 bubbles. Okay. Sometimes they don't write the word bubbles. You just take it as 21, because they already got the word bubble here. Okay, what's the average number of air bubbles? So 21. So there seems to be no unit for this one. And for rate of photosynthesis, it can be bubbles per minute or per minute. 42 per minute. It should be 4.2 4.2 bubbles per minute. Okay, let's look at our results. So distance from Hydrilla species to light source, 10, 20, 30, 40, 50. Number of air bubbles released in 5 minutes. So there are 2 readings. And then you have the average number of air bubbles released in 5 minutes. So add the 2 values and divide by 2, right? And then after that, you calculate the rate of photosynthesis, which is the number of air bubbles released per unit time, okay, or per minute. Now let's go on to some of the questions that can be asked in the biology paper 3 SPM exam. Okay, so practice makes perfect. So let's try answering these questions. Now I've given here a sample result, okay, taken from the previous slide. So I only choose 2 readings for the distance. I mean two different distances, okay, because in the exam they cannot test you so many distances like 10, 20, 30, 40, 50. There's no time to try out all this. Okay, so maybe 2 or 3 values for the distances will be used or will be asked for you to find the responding variable or the results, okay, for you to attempt the experiment. So 10 and 30, these are the readings. There are 2 readings for the number of gas bubbles or air bubbles released in 5 minutes, and the average number, and finally the rate of photosynthesis, which is the number of gas bubbles released per minute. Okay. Now, based on the table of results above, state one observation from this experiment. So try the question first and try to recall what do you need to include, what items you need to include for your observation. Okay, let's look at the answer. When the distance from Hydrilla species to the light source is 10 cm here, the number of air bubbles released in 5 minutes is 22 and 20. Okay, 22 for the first result already and 20 for the second result already. Now, do you fulfill all the criteria for observation? Yes. First, distance from Hydrilla species to the light source. Okay, copy directly from the title here. Okay, and then put in the unit. If it's 10, it's 10 cm, 10 cm. Then that's your MV with the value. And then next is the RV, which is the number of air bubbles released in 5 minutes. All right. And then you must have the values or readings also, which is 22 here, and then 20. Okay, 22 for the 22 bubbles. You want to write bubbles also can. Bubbles for the first result and 20 bubbles for the second result. The unit is not minutes. The unit is bubbles. Bubbles. Bubbles for the first result, 22 bubbles for the first result, 20 bubbles for the second result. Take note, I'm using the raw data. I'm not using this unit, this value, 21, because to obtain this reading, you have to do some calculation. 22 plus 20 divided by 2 equals to 21. It's an average number of air bubbles released. Okay, what you want for observation is the raw data, which is 22 and 20. You write 21 here, you are wrong. You won't get the mark for the value. Okay, so take note, always include both readings if there are 2 readings involved, okay, in the observation, and it must be the raw data. Now let's look at the inference. Okay, inference, sometimes students find difficult to get full marks. Okay, so inference may be 1 or 2 marks. Okay, 2 marks. So state the inference for the observation in 1. Okay, now your observation is based on the distance of 10 cm. And for 10 cm, you have consider a lot more gas bubbles released compared to for 30 cm distance, right? For the distance of 30 cm. So what is involved in an inference? An inference is an explanation for an observation, and we must include the sub-MV, main MV, the scientific explanation, and then relate it to the main RV and sub-RV. So try and write the observation yourself first, and then I'll explain. All right. Now, this is a guideline for you. In this experiment, we have sub and main MV, and then sub and main RV. So all that have to be mentioned. All right. Now here, you talk about the sub MV first. What is the sub MV? It is the distance of the light bulb from the hydrilla, correct? Then is the distance short or long? The distance is short, right? It's shorter compared to the other one. So how does it affect your main MV, which is your light intensity? Okay, then what does the light intensity, what is the effect of that light intensity, which is either high or low, upon the main RV, which is your rate of photosynthesis? Okay, so you must explain the biological effect, if possible. And then lastly, that's why main RV will affect the sub RV, which is your number of gas bubbles released. So can you see the sequence here? Sub MV affects the main MV. Then main MV will affect the main RV. But try to give the biological effect of the main MV on main RV. And then main RV will affect your sub RV reading. That's the reading. Okay, try this question first, and now and then we are going to look at the answer. Right, let's look at the answer. Now, sub MV, when the distance from Hydrilla species to light source, you must mention whether it's shorter or longer. In this case, the 10 cm distance is shorter compared to the 30 cm, right? Then what happens to the light intensity? The light intensity will be higher because it's closer. The light is closer to the Hydrilla species plant. So what's the effect on the main RV, the rate of photosynthesis? Now, this is the scientific explanation. More light energy will be used for photolysis of water. Okay, that is the biological explanation during photosynthesis. All right. So what more water molecules will be broken up into the hydrogen ions and hydroxyl ions, right? Then the process of photosynthesis will increase, right? So this will increase the rate of photosynthesis. So this is your main RV. See, main MV affects main RV. The rate of photosynthesis increases, all right? And finally, that's why when we do this experiment, at 10 cm, you will see that the number of gas bubbles released is more. Okay, so more air bubbles or oxygen gas is released. Okay, so you have mentioned all the MV and RV in this experiment, and you have linked them together very in the right sequence to show the effect of the sub MV finally on the sub RV. Okay, so this is the explanation. Main MV affects the main RV, and how does it affect main RV? Must mention. So this is a super duper complete answer to get you your 2 marks or 3 marks. Now state the 3 variables and the methods to handle them. Okay, so manipulated variable, method to handle it, and so forth. Now you can fill up this table first, and then we are going to discuss the answer. Normally they will not ask all the variables, right? They will choose 1 or 2, and then method to handle them. Okay, let's get started. Now, manipulated variable, as we have discussed earlier, is of course the distance of the bulb from Hydrilla species, or even the light intensity. Okay, so you can write light intensity also. Now, method to handle. Put the light bulb at different distances. So this word different must be there. From Hydrilla species, that is 10 cm, 20 cm, 30 cm, 40 cm, and 50 cm. Note that you must have the word different when you are talking about method to handle manipulated variable. Some people use the word use different distances, but it doesn't sound very correct. Use different distances. So I put the light bulb at different distances for Hydrilla. Then always give the values for the MV. Okay, all the values. 10 cm, 20 cm, 30 cm, 40 cm, and 50 cm. Write this down. It's very important. Okay, all the values that you use for the MV. Now, the responding variable is the number of air or gas bubble sets that are released in 5 minutes. Okay, take note that I'm using the sub RV, not the main RV, which is the rate of photosynthesis. Because if you write the rate of photosynthesis, you have to mention that you have to calculate the rate by using the formula number of gas bubbles divided by the time in minutes. So it's easier to explain the sub RV, which is the number of air bubbles released in 5 minutes. So here's our answer. To handle the sub RV, count and record the number of air bubbles released in 5 minutes by using a stopwatch. You need a stopwatch to time 5 minutes, right? While we are counting the air bubbles. Okay, now your sub RV, please write it as you see it in the question paper. Don't give, let's say the question paper say number of gas bubbles, then you write number of gas bubbles also. It depends. Or if the question says the number of bubbles alone, then you write that. Okay, now here let's assume that it's number of air bubbles like in your textbook. Okay, so count and record the number of air bubbles released in 5 minutes by using a stopwatch. Our keywords here are count and record. Okay, because you have to count. Now sometimes it is measure and record, weigh and record, or it could be observe and record, depending on what your RV is. In this case, it's counting, just counting the number of air bubbles. Okay, so remember to write the word count and the word record must be there. And then you must state the apparatus used. In this case, a stopwatch is used. You have to time 5 minutes while you're counting. Okay, now fixed variable for this experiment is concentration of sodium hydrogen carbonate solution. Not just sodium hydrogen carbonate. Remember this word solution. Okay, because it's a concentration of a solution, not of sodium hydrogen carbonate. What is this? Is this a solid? Right? So it's not, it's a solution. So, or the type of plant used. So how to handle the variable, the fixed variable here? Now we must fix the concentration of sodium hydrogen carbonate solution at 0.2%. Okay, now that is if your fixed variable is written as concentration of sodium hydrogen carbonate solution. Then you write fix the concentration of sodium hydrogen carbonate solution at the value, a certain value. In this case, it is 0.2%. So the word fix must be there. And then another one, fix the type of plant used, which is Hydrilla species. So state the species of the plant. You must state it. Okay. If they use Elodia, then you say Elodia species. So it depends on the type of plant, the species of the plant that's used. Next, the hypothesis for this experiment. Very simple. The higher the light intensity, the higher the rate of photosynthesis. Note that I'm using the main MV, the light intensity, and the main RV, which is the rate of photosynthesis. Okay, this is the proper hypothesis. So can we write about the shorter the distance between the light bulb and Hydrilla species, then the more the number of gas bubbles released. Can we write that to be the hypothesis? Well, it's not so good because it's not the main MV and RV. It's the sub MV and RV. If you write about the distance between light bulb and Hydrilla species and the number of gas bubbles produced. So stick to the main MV, which is the light intensity, and main RV, rate of photosynthesis. Unless you're not sure, then you can write both down. But this is the better hypothesis, as you can see from the textbook. Now, based on this experiment, so remember, for the hypothesis, try to write the main MV and main RV. Now, based on this experiment, state the operational definition for photosynthesis. So remember, operational definition is the definition that is obtained from the experiment, related to the experiment. Okay, and our tip here, memory tip is DORAM, DORAM, D-O-R-R-A-M. Okay, now you can stop the video and then pause the video and try to write your hypothesis, your definition. Don't cover up your phone for a while and try and think of your definition for operational definition for photosynthesis. Okay, let's look at the answer. Photosynthesis is the process in which Hydrilla species produces organic food substances using light energy and is shown or measured by the number of air bubbles released in 5 minutes. The rate of photosynthesis is affected by the distance of Hydrilla species from the light source. Of course, this is definition is quite long, but they give you enough space to write. Okay, and if you write beyond the lines, 1 or 2 lines below is okay. They won't penalize you for that. So here, remember DORAM for operational definition. D is for the textbook definition. You incorporate it into the operational definition. So photosynthesis is the process in which a plant produces organic food substances using light energy. Okay, and then O is for organism. So we have to mention it is Hydrilla species. And then R here is for the sub-RV. So it is measured or shown by the number of air bubbles released in 5 minutes. And then A is for affected by. So the rate of photosynthesis, okay, is affected because the definition is for photosynthesis. So you can write the rate of photosynthesis is affected by the manipulated variable. M is for manipulated variable. The distance of Hydrilla species from the light source. Okay, the sub-MV. Or the rate of photosynthesis is affected by the light intensity. Here I put the sub-MV, the distance of Hydrilla species from the light source. Okay, so with that you should be able to get the full marks. Make sure you state the organism, the actual definition, but then you have to incorporate the sub-RV and the sub-MV inside. So the rate of photosynthesis is affected by, you must mention that's the A, affected by, RV affected by MV. So the next question is to draw a table to show your results. Now we have discussed this just now, okay, earlier. So in the table, usually your sub-MV index for the experiment is written on the left side. And then the sub-RV are written here. And finally, if there's a main RV that you have to calculate, okay, it will be at the end here on the right side. All right. So distance from Hydrilla species to light source, the sub-MV. Now we write down 10, 20, 30, 40, 50. Okay. And then in the experiment was the number of air bubbles released in 5 minutes. There are 2 readings, right? So write down both readings. And then average number of air bubbles released in 5 minutes will be at 22 plus 20 divided by 2, it will be 21. Okay. So all these are the average readings. And then finally, you may be asked to calculate the rate of photosynthesis. As we said, it is one way to calculate is to divide the number of air bubbles by the time, okay, in minutes. So since there are 21 gas bubbles released in 5 minutes. So for 1 minute, you have to divide 21 by 5. Okay, so you get the reading for every minute. What's the reading for every minute? It's 4.2. Okay, 4.2 air bubbles released per minute. So that's your rate of photosynthesis. Okay, and for the last part here, I usually show the calculation, how we get the rate of photosynthesis. Okay, it's better to write down the calculation. Now take note that all these titles here must follow the question and what the question has stated. Don't simply write your own titles. You must copy directly from the test paper what the titles are. Okay, so in this way, you're like a good copier, a good copycat to copy directly from the table of results. Okay, not to copy somebody's answer, but to copy directly from the question paper. Okay, so pay attention what the question paper is asking you to copy, to write down for the titles. And remember your units also. Unit for distance is cm. Unit for average number of air bubbles is actually air bubbles itself. It's not minutes, it's air bubbles. Okay, 21 air bubbles, 16 air bubbles. So if you want to write bubbles, you can, but actually it's really mentioned number of air bubbles. So that's why we don't need to write any units. Okay, but for rate of photosynthesis is number of air bubbles per time. So the per minute must be there. Okay, that means 4.2 bubbles per minute. So in the exam, you may be asked to draw the line graph or a bar chart. Now let's look at each example. Line graph. So you may be asked to plot the average number of air bubbles released in 5 minutes against the distance of Hydrilla species from light source in cm, or even the rate of photosynthesis against the distance of Hydrilla species from light source. So here I choose average number of air bubbles released in 5 minutes. Okay, so to plot the graph, first of all, let's see what are the marks for. You must draw the axis, you must draw the line for the vertical axis and horizontal axis, and then write down the titles, okay, based on the question. The titles for the RV, this is the RV, against the MV. The MV is the one that is on the horizontal axis, okay, which is the distance of Hydrilla species from light source in cm. So copy the titles exactly from the question that's given to you and put in the units, right? Now for average number of air bubbles, it can be in, you can write back brackets bubbles, that is the title, okay, like 5 bubbles, 8 bubbles, and so forth. And then make sure your axis is uniform, okay, and try to draw the graph as big as possible, maybe using up at least 3/4 of the space of the graph. Use up 3/4. If you draw a tiny graph at the side here, then the size of your graph is proportional to the marks. Small graph, low marks. So at least 3/4 of the page must be used up for the graph. Now next, the points must be correctly plotted. So I base it on the table just now. All right, at 10 cm, 21 bubbles were released, and so forth. Okay, down until the last one. Now you can see here is a straight line. So actually, in actual fact, you just have to link all the points together, draw by freehand. Okay, you cannot use a ruler to draw. If the line doesn't come out to be a straight line, then you cannot use a ruler to draw. You just have to join all the points freehand and make it as smooth as possible. All right, then never extend beyond the points given. Don't extrapolate downwards or upwards. Remember that if you extrapolate, then you get it wrong. Okay, bye-bye. Now, next. So 1 mark for points correctly plotted and 1 mark for the axis, okay, vertical and horizontal axis with the titles and units. And then 1 more mark is for the shape of the graph, and the graph must be, if it's a curve, then you must join it smoothly, freehand, okay. Now, what if it's a bar chart? Okay, now let's look here. So for this bar chart usually involves a few values for the manipulated variables, like maybe 2 or 3 only. Cannot be so many. Usually it's not so many. And then the time is too short for you to find out the RV for many different MV. So usually 2 or 3 the most. Now, if it's a bar chart, then you have to, again, make sure your axis has the titles, the units, like cm, and the scale must be uniform, okay? And then next, when you plot the points, make sure the points are correctly plotted. And lastly, okay, like for example, at 10 cm, the reading is 21, right? And 30 cm, the reading or result is the number of air bubbles released is, the average number is 11, okay? Now, then your bar must be the same width and separated. Now, don't draw the bars stuck together because that's not a bar chart. That's not for bar chart. For bar chart, the bars must be discrete or separated, okay? And also the width of the bars must be same. Cannot draw a thick bar here and then a thin bar here. All right. So I must have correct shape of the bar chart and of the bars, and make sure that you have a pencil that's quite dark. Can draw the dark lines to outline because it may be covered by the. It may not be clearly seen if your graph paper is too clear. Then may not be able to see the two bars. Okay, make sure the two bars stand out and can be seen. And don't shade the inside, don't use some shading to color or dirty the bars inside. All right, just draw like that and make sure the line is clear and can be seen. The bar can be seen. All right, now that's another mark for the shape of the bars. Make sure the bars are separated, separated by a space here. Now for the graph, please remember that you have to read the question carefully if you're asked to draw the graph. If they ask you to draw the graph, they may not ask you to write to plot average number of air bubbles released in 5 minutes against distance. They may ask you to plot a rate of photosynthesis against the distance. Okay, so you follow the question and do accordingly, and your titles must follow the question. Adapt well to the question. You must adapt well to the paper, what it wants you to write, and not what you have learned in the class. All right, or what you have listened to from the teacher. Right. So follow the question and write exactly what the question wants you to do. Write down the titles according to the question paper, or questions in the question paper. Right, next we come to prediction question. Prediction question. Repeated with a distance of 10 cm between the bulb and Hydrilla species, but using 0.4% sodium hydrogen carbonate solution. Predict the number of gas bubbles released in 5 minutes, and then explain your answer. Okay, so for prediction question, you must look at the conditions, highlight or underline the conditions, the new conditions, okay, for photosynthesis, and compare with the old one. The old one is the original experiment. So in the original experiment, at a distance of 10 cm between light bulb and Hydrilla species, the plant was also immersed in 0.2% sodium hydrogen carbonate solution. Now you change it to 0.4%. Now at 0.2% sodium hydrogen carbonate solution and a distance of 10 cm, refer back to your table, the number of gas bubbles released was originally 21 gas bubbles. 21 gas bubbles released in 5 minutes. That's the average. So now you are changing the concentration of sodium hydrogen carbonate solution to 0.4%. That's an increase in the concentration of this solution. Thus, what will happen? More carbon dioxide will be supplied by the sodium hydrogen carbonate solution, right? And then this will increase the rate of photosynthesis because carbon dioxide is a substrate for the process of photosynthesis. It's used in photosynthesis. So that means the number of gas bubbles released in 5 minutes will increase and be more than the original amount, which is 21 air bubbles, okay, for distance of 10 cm. So you got to refer back to your table and see the original condition. Then you write like this, right? The number of gas bubbles released in 5 minutes, okay, follow the exact statement in the question. Must copy it down. Okay, so to get marks for this section, one thing you have to do is to copy exactly what is stated in the question, and don't change it to, let's say, instead of number of gas bubbles released in 5 minutes, you write number of bubbles only. You don't mention 5 minutes. It's not correct. So you better mention the whole thing. Number of gas bubbles released in 5 minutes, it's more complete, will be more than 21, because 21 air bubbles was for 10 cm and using 0.2% sodium hydrogen carbonate. Okay, you have to refer back to the table. So now you just increase the concentration of the sodium hydrogen carbonate, you're increasing the concentration of carbon dioxide. Okay, so that means the number of gas bubbles will be more than 21 or 21 gas bubbles. Okay, the unit can be bubbles. Right, how to explain this? So here we usually they will test you about the other factors needed, the other conditions needed for photosynthesis. So here, what is the factor that's involved? It's no longer light intensity, but rather concentration of carbon dioxide. Okay, that is available to the plant, the carbon dioxide that's available. So explanation must focus on the factor, that is concentration of carbon dioxide. Right, 0.4% sodium hydrogen carbonate solution increases the concentration of carbon dioxide supplied to the plant for photosynthesis. Right, supplied to the Hydrilla species. So with more carbon dioxide is reduced in the light independent reaction to form glucose. That means more products are formed and more oxygen gas is released as a byproduct. Right? Now this is the scientific knowledge that we can write in. All right, that may be needed. All right, so try to write it in. If you can't remember, then you just continue with what you cannot figure out what is the effect of carbon dioxide. Okay, on photosynthesis, then you just continue the third one. Thus, with more carbon dioxide, the rate of photosynthesis increases, and this will cause more oxygen bubbles to be produced as a byproduct. That's why the number of gas bubbles becomes more than 21. Okay, so it's good to write down a scientific fact if you know about it. Okay, but you must mention that the rate of photosynthesis, your main RV, increases. Okay, and that's why more gas bubbles are produced. Now here's another question, which tests another factor, right? Which is about another factor. The experiment is repeated at a distance of 10 cm between bulb and Hydrilla species, but the boiling tube with Hydrilla species is immersed in a water bath at 50 degrees Celsius. Predict the number of gas bubbles released in 5 minutes and explain your answer. Okay, 50 degrees Celsius is high temperature, all right? Because the optimum temperature for the Hydrilla species to carry out photosynthesis is around 25 to 30 degrees Celsius. Okay, so explain your answer. So first you have to predict the number of gas bubbles released. So again, you copy this down. Number of gas bubbles released in 5 minutes will increase or decrease. Now, temperature is 50 degrees Celsius, is too high, right? So what happens to the enzymes? Enzymes of photosynthesis will be denatured. Don't write damage, because the scientific term is denatured, right? So that means the number of gas bubbles released in 5 minutes will be less. Okay, now less than what? So again, we refer back to our table at 10 cm, and temperature of the temperature was room temperature when you carry out this the original experiment. Okay, room temperature. So now you have increased the temperature. Now at room temperature, the number of gas bubbles released in 5 minutes was 21. Okay, so now you increase the temperature, the number of gas bubbles released will be less than 21. Okay. Next one, explain your answer. So again, you look here, what you try to find out what is the factor that's involved. It's no longer light intensity, okay, compared to the old experiment just now. It's no longer concentration of carbon dioxide, but it is the temperature, right? Okay, so you have to explain what's the effect of high temperatures on the plant, okay, on the photosynthesis of the plant. At a high temperature of 50 degrees Celsius, the enzymes used in photosynthesis are denatured. Okay, so photosynthesis requires the use of enzymes to catalyze the reaction of photosynthesis. So the enzymes are affected by temperature and they are sensitive to temperature changes. So if the temperature is too high, the enzymes will be denatured. Okay, that means their original structure is already changed, is destroyed and changed, and the polypeptide chains will unravel. Okay, and the active site will be destroyed in the molecule of the enzymes at high temperature. So the catalysis will be slowed down, catalysis for photosynthesis. So the rate of photosynthesis decreases, all right, due to the fact that the enzymes are denatured and less oxygen bubbles are released as a byproduct. Okay, so the number of gas bubbles will be less than 21. So let's look at another question, which is on classification. Okay, this is the last question. Now, a group of plants were exposed to the following conditions in a greenhouse. Okay, the enclosed greenhouse, all right, which has glass walls to allow sunlight to come in. So, one condition at a time. Classify the conditions into the two categories below, namely favorable conditions and unfavorable conditions for photosynthesis. Okay, so all these conditions are tested separately, right, using a group of plants. Now, which one will increase the rate of photosynthesis? That will be the favorable condition, right? And the situation or condition which causes the plants to have decreased rate of photosynthesis will be the unfavorable conditions. Okay, so classify these conditions into the two groups. All right, so try this question first. Right, let's look at the answer. Which one is favorable? Now, look at the titles carefully, okay? So you got to read the question and don't assume anything as though it is like the textbook or the slides here. You must read the question and see. Here they put unfavorable conditions first, and this one is favorable conditions, all right? So state the unfavorable conditions here, which is green light and dry soil. Okay, now green light is not absorbed by the plant, right? So it's reflected, so the plant will not have enough energy for photosynthesis. And then dry soil will cause the plant to have a lack of water. Water is a substrate for photosynthesis. So these two conditions will cause the unfavorable conditions that will cause the rate of photosynthesis to be reduced, to decrease. Okay, whereas these are favorable conditions. Okay, on this right side, temperature of 28 degrees Celsius. This is the optimum range. The optimum range of temperature for the highest rate of photosynthesis is around 25 to 30 degrees Celsius for most plants. Okay, 25 to 30 degrees Celsius. So this falls within the range of the optimum temperature. So this is a favorable condition that will allow the photosynthesis rate to increase. Blue light and red light, they are both absorbed by the plant, mostly absorbed by the plant, because the chlorophyll absorbs the blue and red light well. So that will provide energy for the plant cells to carry out photosynthesis and for the photolysis of water. Okay, right. So this is the way that we can write the get the answer or write the answer for classification question. So here's an alternative setup, apparatus setup for this experiment to investigate effects of light intensity on the rate of photosynthesis. Now, in this alternative setup, the light source is still there, and then here we have an inverted funnel and an inverted test tube. Okay, so the plant is placed inside the funnel, and this whole apparatus is placed in the 0.2% sodium hydrogen carbonate solution. All right, then the plant is also weighed down with a clip or plasticine, and it will produce oxygen gas in the presence of light during photosynthesis. So the air bubbles, oxygen bubbles can be easily counted here. Okay, and the test tube that is inverted over the filter funnel will collect the oxygen gas produced as a byproduct of photosynthesis. So in the next video, part 2, we will discuss the effect of 1. Carbon dioxide concentration, 2. Temperature, 3. Color of light on the rate of photosynthesis. So stay tuned for the next video. Goodbye for now, and all the best for your exam.